we will go into biomolecules, looking at basic structure of living things, starting with cell. When we start with the cell, our goal here would be looking at some functional characteristics, so which will serve a little bit on the engineering side, or how can we quantify those processes or those things that happen inside a cell. Understanding those things are important because when we talk about Bio-Nano, we have learned about nanotechnology a whole lot. We have good idea of how we can make things smaller. We need to get a little bit understanding of what may be of our interest to study, what we would like to study on the living side. So that starts with the cell. So and cell is the basis of all living things. Everything that is alive is made of atoms and molecules, and then they come together and they form, in living things, they form cells. And these cells have different size scale. These and subcellular things are at different size scale as well, all the way from microscopic to macroscopic. Now, many of the things we will discuss would be at cellular level or below cellular level. And the reason is because that's the basic functional unit of life. It is it is what makes any organism, these are what give any living organism their basic fundamental properties. Now, there are two types of cells in nature. One is called a prokaryotic cell, which is a complete organism. It is self-contained. It doesn't have a clear nucleus in it, and the nucleus, or it has some functions like nucleus, but they are not contained in a further inside a membrane within the cell. And their internal subcellular parts or things are also not contained within walls or things like that. So there's no clear-cut partitioning of different things in a prokaryotic cells. And these cells are they range in different sizes. They can go as many as few micrometers. A good example is bacteria, right? So they're self-contained, single cellular organisms. Eukaryotic cells, on the other hand, are cells which have very well-defined boundaries. And within those cells, the subcellular entities have very well-defined entities and membranes themselves, which makes us to define the things, how they are happening within those cells. And that's where we have the capability or potential to do something, whether we are measuring, whether we are trying to change. So this is a picture of a bacteria, if you can call it. What it's showing is a simple representation. In the place of a well-defined nucleus, there is nucleoid. And that nucleoid contains the genetic material for the cell. There is few more things: cytoplasm, ribosomes, flagellum. So we'll see many of these things in eukaryotic cells as well, the cells which we are made up of. And there are, for these cells, there is this long thing, flagellum, that's coming out, that helps them move, that helps them propel the cells forward or whatever their function is. Now, these are cells which are self-contained. Now, when we talk about uh, any cell, they perform many things in whichever way. Within those cells, and some of the things we'll look at very closely. Some of the things, just names are there, don't worry about it, don't get too much terrified. But I know when we talk about nanobio, or when maybe for KPR also, when you were looking at their applications, we probably have not talked too much about the bio side of things, but we will. The goal is to give this introduction this week, that we should be able to, we should not be scared of looking at biological entities. Or if you read a paper where they have done some biosensing, we should be able to get some idea of what does it mean, or where does it fall, or where can we look up and see what is the importance of that kind of thing. In these cells, there's this thing called plasma membrane, which is basically containing the cell together. We don't consider it as an organelle, but we list it as an entity that it belongs to a cell, because plasma membrane carries out many important tasks, many important things in our uh, cell functions. This is the interface with the outside world. The plasma membrane provides many pathways, many connections, many openings with the outside world. So that's why we need to put it, and we'll look at a little bit closely at what happens in plasma membrane, how is it formed, and what are different things that go on. Now, this list, if we map it, so we can see some things right away. So there's a distribution of things inside a cell. There are organelles, small parts, which are, some of them are providing support to the cell itself, like maintaining the framework of the cell, keeping the cell in very specific shape or very specific form. And some of them are doing things just within the cell. Some of them are getting some molecules from nucleus or from outside of the cell to carry out their functions. So there is a whole machinery that's working there. As I said earlier, our goal in this lecture is to look more at cell like a machinery, and what are different things or some important things that we as engineers or scientists have want to interface with nano, or want to understand, or want to do something about it if things don't go the way we expect them to go. So this is just the general layout, how things are. So it is mostly fluid, mostly water, and these things that we look, they are all suspended in water, right? Now, you have to understand, pure water is a good insulator, but water, the way we have, has lots of salts in it. Salts become ions when they are in a polar solution, so they are available for movement. If you apply potential, we can move charge, and that's what that's what a cell wall, or we call them pores, nuclear pores or cellular pores, small holes, they push ions across. And that's a machine, right? So if you think about it, there has to be potential applied to push ions, charge entities or charge bodies anywhere. Now, let's take a look at plasma membrane first. So plasma membrane, the thing that makes the outside boundary of a cell. It is not a hard boundary. It is not a very rigid uh, boundary, but it is made up of very small molecules. It is built up out of number of molecules. Two things I want you to see first. I want you to see those red balls, which are like all stacked together. And every red ball has two yellow legs. These are the fundamental building blocks of this membrane. These are the building blocks of a cell wall. They come together to form this long wall. And then there are things which are part of that membrane. We'll talk about some of those. Right now, I want you to just focus that plasma membrane is the outermost covering of a cell, and it separates it from other cells and from the outside environment. And this red and yellow thing, these are called phospholipids. So, and if you look closely, there are two of those. So one phospholipid is on one side, and another phospholipid layer is on the other side. So we call it bilayer. We call it phospholipid bilayer. So this plasma membrane is made up of phospholipid bilayer. So it has few functions. Number one is it maintains the internal equilibrium of the cell. So by very selective entry and exit of molecules and ions and whatever needs to come in and out. So it's like border security, right? So not everything can go inside, not everything can go outside unless it's regulated by plasma membrane. Now, it also means that it maintains a stable internal environment, constant internal environment. Now, when we say or you think about exit entry like border or airport, or it regulates the things that go across cell wall. So it regulates, they say, transmembrane transport. So everything that goes in or out has to be regulated, or the plasma membrane has to ensure it's the right thing or it doesn't. Now, it also prevents essential molecules from the cell to diffuse outside. Many times it would happen that there is different concentration of something outside or inside. If there is different concentration, molecules are supposed to travel by diffusion, right? So from higher concentration to lower concentration. So that's another job that of the plasma membrane, that it would not let uh, diffusion or any mean to occur for those loss of essential molecules that are required. So hence, again, maintaining a stable and constant internal environment. Another important thing that plasma membrane does is cell signaling. And that cell signaling can be between cells. Cell signaling can be from similar type cells, or it can be from different types of cells. But as engineers, we know signals, right? We know signals. You transmit a signal, and we think of electromagnetic waves that carry our signals or carry our messages. It's a similar concept here, but here the signal would be in the form of a very specific uh, uh, few or a group of molecules that would be released. Or when you send a signal, you also have to be able to receive a signal, right? Receive a message. Or in that case, that would be the specific molecules on the boundary that would bind to an incoming molecule. That incoming molecule, once received, would be called as the signal has been received. You have to think, and it's an amazing kind of arrangement, and very similar to how we know things in hardcore engineering, that we send signal, we receive signal. In that case, we send uh, electromagnetic waves, or we send a coded message in the form of potential energy. Here also we are sending signal, but here the signals are molecules itself. Now, these receiving antennas, if you can call them, we call them receptors. So they are within the membrane, and they would bind to that molecule coming in. So we call them receptors. They receive those molecules. So they are also part of plasma membrane. Now, when we make drugs or we make different therapies where we say, okay, for example, Panadol, right, or Tylenol in West, so we take those drugs, so they're doing nothing more but finding those cells, either getting inside cell or satisfying those receptors to heal the cell or make it feel like that things are okay. So receptors are major focus of designing new drugs and therapies. Now, these membrane also has protruding molecules sometimes in it, which would be used to bind to other things, or to adhering to maybe other cells, to other membranes, to tissue, whatever is the part of the thing. So plasma membrane plays a major role in how cells are regulated. What we would be interested in, when we get a little bit in Bio-Nano, we'll see that cell receptors or cell-based molecules tell us about either their quantity or their high overexpression or their downregulation is indicative of disease state. That is something we can use as engineers or as making new devices to study or find the state of cell. So that's something which is interesting for us. All right, so it's a new topic. I can imagine many of you might not have gone in depth in these things, but today we would be going into more into bioside and then ultimately making a connection with the nano side. All right, Emma, question.
Sir, so how thin is the cell plasma membrane? Is it in
Smaller
micrometers or
nanometer.
nanometers?
We are talking
So
about nanometer. We are talking about, hold on, we're talking about a few carbon atoms chain.
like it's so we have to make how on average how thick or thin, I
The
mean.
the lipid bilayer,
Yes.
the
The,
lipid
pl-
bilayer, we can,
no, no,
okay.
the plasma membrane plasma membrane
Plasma
actually.
membrane,
Uh,
probably less than less than a micron, even half a micron, probably 500 nanometers.
so we have to make so if we want to make if we want to a bioman's particle to go through it, would we need to make it very small than get
So how do we make things go through this membrane? It's not that we have to push through. It has to use the chemical nature of these things which are floating, which are part of this membrane, like this
okay.
glycoprotein, or there are receptors called epidermal growth factor receptor, EGFR. We can bind to those things and cause a cell to do certain activities that we would want to do. So
Okay.
yeah, and we can coat particles and send those as Trojans. You know, Trojan, like a story where they were
Yes.
soldiers were hiding inside a horse and they thought it's a horse and the soldiers. So there are lots of that's you will be studying these things, that what are different ways how people have sent things inside a cell to do something, to maybe kill
Okay.
cancer cells from inside out, right? To disrupt the function of a cancer cell by introducing external materials, nanoparticles, for example. Oh yeah, these are very, very important questions which, or ideas that we will study and we go forward.
Okay.
Yeah, very good.
Said in your technology, don't we have to think about the toxicity?
So that would be
What?
another thing that if we introduce something, can living systems get rid of those? Can living systems digest those and turn them into less toxic things and get them out of the body? That's also function of the cells. That can we introduce something, can cells manage those or not?
Mm-hmm.
Yeah, so that would be when we get from cell to tissue level. Then we will become more interested in what's the overall effect of whether these are in lungs, whether these are in bladder, whether these are in circulating in our body. So whichever uh, thing we are addressing or targeting with our nanomaterials, those organs, can they get rid of those in reasonable time, or decompose them and get rid of those? Yeah, that would be a direct function of cellular operation, or how cell is managing all that. So very interesting question, very important question, because all of these have to do with the cellular or subcellular operation. Now, we see here these blue proteins. These are all membrane proteins. Now, you have to understand, in nature, in a cell, all the molecules that are formed from amino acids are a bunch of molecules. There are 20 type of those. We call them proteins. Functionally, they're all proteins, but then not chemically, they're all proteins, but functionally, because they have different things they do, we classify them, and biologists then use their names to whatever they do. But as engineers, just remember, all of these are proteins, right? Here we call them membrane proteins because they belong to membrane. You would see some of these proteins are internal. They are only in the inner side of the cell. Some of are they are only towards the outside of the cell. Some are who are going across the cell membrane, right? So there are some proteins which are integral membrane proteins, and they have hydrophobic regions. So now hydrophobic is an important property of living things. Hydrophobic is something that doesn't like water, right? So when there is a hydrophobic portion to a protein, the protein would align itself in a way that it wants to keep that portion away from both sides, because both sides has lots of water. For example, these lipid bilayer, the two yellow legs are hydrophobic. So nature, because of their hydrophobic nature, they come align itself in a way. The reds are facing outward, they are hydrophilic, and they're protecting their yellow parts inside. So both sides have come together, and that has formed a membrane, that has formed the cell wall. Think about it, the beauty of this, that because they have hydrophobic part, or like this integral protein, which is the globular protein, the blue one, the big blue one going across. So why or how it is staying in the middle? Because it might have some internal part which is hydrophobic. So it is right there. It doesn't want to go up and down because the middle part, which is hydrophobic, does not like water. So hence they stay in place. They penetrate until that part that the hydrophobic portion is not exposed to outside of the cell or inside of cell, because they have lots of water there. And then there are some which are called peripheral membranes, peripheral membrane proteins, and they have only hydrophilic portions. So which means they are on the periphery of the membrane. They are okay. They can float around right on the periphery. It doesn't have to be taken inside. These are important considerations as engineers. When we design something, we have to know hydrophilic and hydrophobic parts and how would they interface with living things, or which part of the living thing are we targeting. So do they match in their hydrophilic and hydrophobic nature, whatever the requirement there, right? So these are proteins, different considerations of where they would fall or where they would go. Now, all proteins are embedded in lipids. So lipids is another class of molecules. In this, the format of lipids is something where they are formed by non-polar hydrocarbons. Now, what is non-polar hydrocarbon? These are the molecules which has special properties. So either they are doing energy storage, or they are just making the membrane, or they are just they're harvesting light energy. So whatever their function is, that's what they would do. They would now the membrane also has protein, the carbohydrates and cholesterol molecules in there. So these prevent or do whatever they're supposed to do. Now there's a thing called fatty acid, which are non-polar hydrocarbon, which has a carboxyl group, which is COO. If you remember carboxylic acid, COOH, it's COO, and a hydroxyl group. Hydroxyl group is OH group. When they come together, they form a glycerol. Now, don't get worried about these chemistry terms. The take-home message here is that there are some building blocks which have chemical affinities. So like carboxyl group comes to hydroxyl group, and they form a molecule, we call it glycerol, right? Now, these lipids, the red and the yellow one, these are phospholipid. When they come together, we call them phospholipid bilayer. These are considered the major structure of a cell, right? The phospho portion is hydrophilic, So that goes to the outer side, and it's okay with water and other polar molecules also. The lipid tails are hydrophobic, so they are to the interior of the bilayer, kept away from water or exposure to water, right? So this is just the basic structure of phospholipids. The red thing that was shown there, if you break it down, these are the simple things. There's hydrocarbon chain, CH, 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 right? And on the top, we have a phosphate, which is PO. And on top, we have another group, what we call as choline. So we know exactly what they are made up of. We can go down and we can go close and we can look at their configuration. But that's not very important. What's important is that there are certain parts of molecules which would be hydrophilic, and they would uh, be towards the water side. Some are hydrophobic, they're on the other side. If you look at closely, this is something like this that we would see, right? So hydrophilic heads and hydrophobic tails, which are on the other side, right? Now, if you think about it, if you look at closely, it's a large molecule, right? It's a molecule at the end of the day, nothing else. Atoms and molecules, basic elements coming together. Now, we also define a lot of things in living in nature by what are they doing. So when there is a long chain of smaller molecules, we call it a macromolecule or a polymer, right? So when those polymers or those molecules, they come together and they do something, then we put them into separate types: proteins, carbohydrates, lipids. But basically they're all molecules. Yeah, question.
Sir, sir, while we were talking about bio-nanoparticles, I never asked how feasible is it to make them? I mean, I sort of
Nanoparticles
how,
will get there. Where we are getting into next module is synthesis of nanoparticles. How do we make
so
nanoparticles?
sir, okay, so how so wouldn't it take sort of like lots of money to actually make these polymers and macromolecules?
No, it's very cheap. You can make them in your garage. You can make in a simple lab. If you know the chemistry, if you know what needs to be added up, you can make these. Hydrocarbon CH long carbon chains are easy to buy, easy to find.
What
You
about
will be amazed.
buckyballs? What about
See,
buckyballs?
buckyballs. Okay,
Buckyball, yeah.
so so we have recipes of those. How to make them?
Mm-hmm.
There are very simple recipes or SOPs, as you say, are available of how to make them. But that's not what we are studying today. We are getting inspiration that this happens in nature by itself. All of us. Look at any cell, this is happening there. So that's
Okay.
something which can be translated into synthetic world as well. Right? All right. So many molecules they come together. Sorry, many smaller molecules they come together in long chain. We call it polymer, right? So those individual parts we call them monomers. Why I'm going, these are same concept which you know, but when we read biological literature or when you read nanobio papers, you will encounter these words, monomers or polymers. So you have to understand what does that mean. When these molecules they come together and their molecular weight is more than 1,000, we call them macromolecules. Nothing special, just how they come together and what forms them. Now, these proteins, polysaccharides, which are large carbohydrates, nucleic acids, they all are macromolecules, right? So, looking at or their distribution or their differences in their structure doesn't mean they are not macromolecules. They are all macromolecules or they are all polymers. But what they do is that would identify them. What are they what do we call them, right? So each type of macromolecule performs some combination of some functions, whether this is it's doing energy storage, whether it's doing structural, it's providing structural support to the cell. It may be protecting it to something from coming from outside. It may be doing catalysis. It may be transporting things around. It may be defending cells from disease or from problems in DNA replication. It may be regulating those processes that are supposed to happen and keep cell happy and help it grow and then divide, and then smaller uh, daughter cells start doing the whole thing. They perform functions like movement of cells. And some of them, like DNA, uh, they do information storage, right? So each micromolecule is doing something different from each other. And now it is not that one type of molecule is doing one of these things. So maybe multiple molecules have to work together to achieve some goal. For example, uh, carbohydrates and proteins, they both provide uh, structural roles, and they also provide uh, and protect tissues and organisms. So they may be working together. So there are building blocks of bunch of molecules who do all different things. There's one type of molecule that specializes in information storage. Can somebody tell me which one? Can somebody tell me what molecules do the important part of information storage?
And the brain cells.
Brain cells? No, molecules contain the information. These subcellular molecule or macromolecule who do information storage. I'll give you a guess. They also have an important function of hereditary materials. DNA. Yeah, Ahmed, you got it. So see, now DNA has a very special role, which is to just contain information. So every part is doing something very different from each other. Now, if you look at these macromolecules, it's blowing up in this graph. It's showing that there is 70% water, very small percentage of ions and small molecules, and a huge part is what makes up all these. And out of those molecules, about 1/4 is nucleic acids. DNA is a type of nucleic acid, right? Just stay with me. We'll see why do we call it nucleic acid, or how many types of nucleic acids are there, right? Now,
because it contains hydrogen plus atoms.
Nucleic acid, of course, to be acidic, it has to have hydrogen ions, right? It has to be positively charged.
Yes.
Right. But there's more to it. But we'll see. We'll look at that. Now, each of these macromolecules, right? So remember, I'm using macromolecule, which is I could have called it polymer also. It's the same thing, right? Today, learning a few things. Biologists use these terms very fluidically. We engineers, we call specific names to everything, but in biology they have many. There can be more than one name for one thing, whether they're talking structurally, whether they're talking about functionally. So it depends on the context we're talking. So now all of these have bunch of small groups into it, which we call functional groups. And there's nothing to it. It's just bunch of some atoms that come together, but they come in a way that they form specific shape, and they have some chemical properties. So two things are important for what we call them functional group. They have to have a certain shape, and they have to have certain chemical properties. Now, when you say shape, how do we get shape in a molecule? It may be they fold in certain way. They may have a kind of a compact form that they get into. And that is very specific compact form for that sequence of molecule that they are. That folding is important because if they don't fold in specific way, that's called misfolding. And misfolding is misfoldings of certain molecules are known to be reason for diseases like Alzheimer's disease. It's known now that it's because of misfolding of a protein in brain. So, but stay with me that functional groups are macromolecules which have the some functions which come from their specific folding and their chemical properties. Chemical properties of their monomers, those individual groups that come together. Now, that properties would result into if they are like water soluble or not. Maybe they are not water soluble. That property would be maybe they are hydrophobic, maybe they are charged, maybe they have positive charge. So that folding results into very specific properties. And those properties then make them suitable for whatever kind of reaction there's. Maybe they have to do interaction with another molecule, like we were talking about signal and receptor. So that specific signal, which is a molecule, has been sent. Now that cell that's supposed to receive has this macromolecule that's supposed to bind to it. So it may have certain folding, which gives us maybe opposite charge for it to bind to it. So, but these are the basic properties which come from the functional groups. Now, there are some proteins and carbohydrates that may create long fibrous systems that would give strength to the structure. There may be some, because of those functional groups, they can come together and give structural strength. Some may be long, thin assemblies of proteins that can kind of contract or expand, right? So that results into the movement for the cell overall. So if it cell can go and travel, they're fantastic videos. If you look cell movement, you will find amazing videos where you see cell actually moving. That property comes from some of those assemblies of thin assemblies of proteins coming together because of the functional groups, right? So macromolecules are large and they contain many different functional groups, which gives them their shape and their function. And those could be the function could be they are hydrophobic, could be they become polar, it could be they have charge to it. So very specific properties, right? Which ultimately come into play when the cell is, or whatever a cell is supposed to do, right? Now, think about it. We are talking about everything within one cell because it's a big factory. It's a big machinery that's working. And this diversity gives a very different kind of a range of interactions between macromolecules and hence a range of properties to the cell themselves. So what are some functional groups? And you will see these are nothing but basic chemical groups, right? And they are small. Hydroxyl is OH, aldehyde is CHO, keto is CO, carboxyl is COH. When they bind to a longer chain, they give that property of hydroxyl, for example, they can bind to an ethanol. That becomes an ethanol. So there is this functional group, two Cs. When they bind, two carbon chains, when they bind to OH, becomes ethanol. It can be longer chain, then it will have its own name, whatever it is. When you have aldehydes coming together with just a C, just a carbon becomes acetaldehyde, acetone, acetic acid. So now breaking it, you have to see, instead of organic chemistry from 12th grade, we are coming now from a bio side, from a functional part of that thing. So it's interesting. In organic chemistry, you have learned about carbons, right? And nomenclature of carbon and all that. Here we are talking about functions. That's it. Because these groups are providing specific functionality to the molecules. All right. So far with me, is it too much for today?
Yeah. Could you repeat that, sir? So these are sort of like what interact with sort of like the cells.
They all have different properties, different functions. When they come together, they do different things.
Okay.
If we look at internal mechanisms of cells, many of these functional groups would be acting up and giving those functionalities to the cell. And that's where we want to come in as engineers and build something to see what's wrong or to sense those things. Okay, so we talked about DNA. Where do we have DNA? Where is DNA stored in a cell?
So DNA is sort of in the nucleus, I think.
The nucleus. Yes, you're right. You are perfectly right. So within nucleus, much, not too many things happen within nucleus. It's the largest organelle, but interestingly, very few things happen inside DNA. And the other things, chloroplast and mitochondria, and their size of those prokaryotic cells themselves. So even in that kind of cell, the other type, the bacterian type of cell, the genetic material and protein synthetic machinery is there, and they are put in nucleides. But in eukaryotic cells, our cells, we have a specific kind of a boundary where we duplicate DNA, where we give the genes out, the information out from the genes, and we'll discuss that also. And we make certain properties in that cell. We make that cell do something certain way, right? Like the features. How do I look like? We look like our parents. The color of skin, the color of our eyes, a lot of things, they come from there. Now, there is many proteins are there which are encoded by the DNA, which is saved in the nucleic acid, in DNA, but they're not made in the nucleus. We think about it. You have a book, huge big book. You want to study a chapter. You take it to a photocopy machine. You just copy 6 pages out of it, and you read those 6 pages, and you do not touch your big, heavy book, right? That's very much like that. So we have this whole bunch of DNA, 3 billion base pairs, 3 billion bases. But one gene may be 12 bases long, or I don't know, 200 bases long. Those 200 bases would just open up, give that information to another molecule. That molecule will come out of nucleus and translate into a protein, which it was supposed to express. These are technical words. Gene expression. The expression happens outside of nucleus. So the nucleus is itself surrounded by a double membrane, right? We call it nuclear envelope. Now, in the nucleus, we have the DNA, but whatever activities it has to do, some of them are made outside of DNA. Some are available inside. But there is an activity where there are holes in the nuclear wall, in the nuclear envelope. They're called nuclear pores, very small, nanoscale pores. So these pores are used to transfer substance across the nuclear membrane. Now, let's think about it. A whole cell is protected by a membrane, and then nucleus itself is now protected by a membrane. And within the nucleus is DNA, the genetic information. I don't know, you can call it Lohe Mahfuz, the safe place where the whole mechanism of that cell is stored. right? And then there are some molecules that come together. There's a thing called chromatin. So chromatin uh, becomes active to, before cell division, it changes the whole DNA into this kind of shape that's shown in this figure. We call that, this shape of DNA when before the division is called chromosome. So chromosome is nothing, the same DNA which would be there, but now cell is ready to divide. It would, there will be a thing, chromatin will be made. So DNA will combine with some proteins to form this structure, this entangled structure. And chromatin will aggregate this structure called chromosome. And then division happens. We'll probably not go into division, but we are more interested in where things go wrong and cells become diseased. So we can do something about it, right? Nanobio, that's where you want to come in and look at these things. All right. Now, within the nucleus, there is a bunch of things. Again, two layers, outer membrane, inner membrane, and there's chromatin, nucleolus, there's envelope, pores, and everything in that soup, which they come together, and before division, they break apart. But we want to study a little bit more on what is DNA made up of, and why it is important, and why we would want to study DNA and RNA and its expression. Right.